working. Okay, welcome everybody. Sorry about the glitches. It's always something. Now I'm going to try to share my screen. Hopefully that's going to work. Okay, great. Start. Okay, great. All right. So welcome everyone. And today in our webinar, we're just going to share with you some new updates from the team about new data and new features in the knowledge portals. Let's see. Okay. So we'll first talk about some new data sets that we have queued up for our upcoming release, which is going to be in a couple weeks. And then we're going to talk about some updates to the gene page. And I'll go over some of them, and then Annie will talk about the tissue-specific gene expression viewer, which she has implemented. And then I will give a sneak preview of a new feature or updates to an existing feature, the Gene Finder, which is those updates are going to be in our upcoming release. And then Noelle will talk a little bit about future directions. Okay, so first of all, I wanted to talk about new genetic association data sets. And so our team who are responsible for loading all these genetic association data sets are Chang, Yue, Patrick, and Ryan. So Chang and Ryan are computational biologists, Yue is a bioinformatician, and Patrick is our back-end software engineer. And together, they've developed a system that has really allowed us to just way ramp up our loading of data sets. And so for this upcoming release, we're loading 48 new data sets that representing associations for 79 new phenotypes. And we're also loading a lot of credible sets, which I'll come back to in a minute. So for this release, we decided to focus on some of the areas, the organ systems that are not as well represented in the association to function knowledge portal, like the digestive system, for example. So Chang and Yue went looking for data sets that have these phenotypes in the GWAS catalog. We're now importing a lot of data sets from the GWAS catalog, and we've also looked for these just independently in the literature. And of course, our colleagues also contribute data sets. I see we have a question already. Let me just take a look at what that is. Okay, let's see. Eva has some questions about this platform, which I think we'll have to answer later. But we have a raised hand also. I don't see that. Mackenzie, do you see a raised hand?
Yes, so I just asked that person to unmute.
Okay.
Okay,
right.
I guess not.
Okay. All right, well, you can come back to us with your question later. So in this new release, then the Association to Function Knowledge Portal, which, if you remember, is the portal that has all of the data that we have, will have a total of 595 new data sets and 668 phenotypes. And so the other portals, such as the Common Metabolic Diseases Knowledge Portal or the Type 2 Diabetes Portal, all of the disease-focused portals, they share the same data that's in the Association to Function portal, but just particular slices of it. And they're aimed at people, at disease-specific research communities who want to focus on those diseases. So I just want to run through a few of these data sets. I'm not going to show you all 48, but I just wanted to mention a few of the new data sets in some of the new areas that we're going into. So, for example, one area we're trying to get into is the reproductive system. And so we've added some data sets for new phenotypes for pregnancy and reproductive traits. And we're continuing to do this, but these are some new ones that we've just gotten. Some of these are very recent studies. The digestive system, as I said, we're trying to increase the number of data sets we have for that one. And it is somewhat of a less studied area than some others, but we're looking for these data sets. And we've added a couple more publications for irritable bowel syndrome, another one for inflammatory bowel disease and Crohn's disease. and then some phenotypes that are new to the portals, an association study for diverticular disease, gastroesophageal reflux, and Barrett's esophagus. In the area of the nervous system, we've recently added some large data sets for Alzheimer's and Parkinson's. And in this release, we're adding a couple more for ALS, and also a data set with epilepsy phenotypes, which we have not had previously. For ocular traits and diseases, we have, I couldn't even list the data sets because we've added a lot of them. So I'm just showing the new phenotypes that we have. So we have some data sets with associations for ocular diseases like AMD, cataracts, and so forth. And then we have also some data sets that have a lot of ocular traits. For example, the thickness of different layers of the retina or the layers of the photoreceptor cell. And finally, we have a really interesting new study that's quite different from anything we've had before. It's a study of associations for response to drugs. So this is a study from colleagues here in Boston at Massachusetts General Hospital and also at the Broad Institute. And they took a group of participants and gave them a dose of a sulfonylurea drug, glipizide. And then at a prescribed time period after, they gave them metformin, and they did a glucose tolerance test. And all through, they were taking samples and measuring, glucose and insulin and so forth, all these glycemic measures. And so, and of course, they genotyped these people and then did GWAS. And so this has resulted in a lot of new phenotypes, all of which have to do with how these people responded to these drugs. And so working with the authors to understand these and categorize them, we have two new groups of phenotypes, metformin response and sulfonylurea response, and all these phenotypes are under them. So hopefully we'll be getting more response to drug associations. I think this is a really interesting area for people working on diseases. Okay, so credible sets. Excuse me. As you know, credible sets are just sets of variants that are put together by researchers who've done a GWAS, and these sets represent the group of variants that is likely with some defined probability to include the causal variant at an associated locus. And so they're just sort of narrowed down most likely variants that are going to be important. And so these are really important for prioritizing variants and genes in GWAS loci. And they're visible on our variant sifter tool, which is that tool that allows you to stack up genetic associations, credible sets, epigenomic annotations, and so forth to narrow down the genes and variants of interest in a genomic region. So we've up till now, we've only had 11 of these sets, I think. I'm sorry, we've had 40 of these sets for 40 traits, but that number hasn't changed for a long time, and we're now making a much bigger effort to get these. So Yue, our colleague at EBI, has been searching the literature, developing automated literature searches to find papers that have these sets, and then Chang has been looking at the papers and finding the ones that do have credible sets that we can use, and formatting them and so forth, and uploading them. So with this release, then, we're adding credible sets for all these phenotypes, and we can now really ramp up this process. So we should be able to have credible sets. Our goal is to have credible sets for all phenotypes that are in the portals. Okay, and finally, another type of data that we're adding is gene level associations. So you might remember that on the gene page, we have had for a while a display of rare variant gene level associations for a gene. And previously, these were just across 24 phenotypes. So they were based on exome sequence association analysis that was done by Jason Flanick and colleagues for type 2 diabetes and 23 cardiometabolic traits. And so that for a long time, those were the associations we had here. recently, in our last release, we added some unpublished gene level associations that came from a study from Sean Juergens and Patrick Eleanor, looking at UK Biobank participants. And we added many more phenotypes from that study, gene level associations for those. Now we've taken some gene level association scores from I don't know if people say GeneBAS or GeneBase. If anyone on the call knows, let me know. But either way, it's a great resource, and it contains exome sequence analysis of UK Biobank participants, and it has gene level scores for many, many phenotypes. So we've taken from GeneBase the gene level association scores for 189 phenotypes that match those that we currently have in the portals. And those are being added to this display as of the upcoming release. Mackenzie, you'll just let me know if there's a question that I should answer right away. I hope. Okay, good.
There is one question, which is, why isn't the portal team calculating their own credible sets?
Well, we have used in the past a calculated credible set method, but it's just not as robust as the methods that people use. But that's a good point. I mean, I think there are robust methods, and we could implement those, and then all the credible sets would be consistent. Yeah, I think probably the answer is we just haven't gotten to that yet. But yeah, if you think that would be a valuable thing, I think that's good to know. Okay, and now we're going to move to some demonstrations, and we'll look at the gene page, and we'll look at work by our front-end team, who are DK and Annie and we. And then we'll also see some of the huge calculator scores on the gene page, this was a method that was implemented by Oliver. So I'm going to switch. Let me, whoops, escape and get myself a web page here. Okay, so first I'll show you, just for context, what's in the portal today. So we're reorganizing the gene page a little bit. So here, this is the current production portal. And on the gene page, when you scroll down, there are 3 displays that are really similar to each other, and they have related information. So on the top, there is the huge scores. And so these are scores from the Human Genetic Evidence Calculator. And as I said, Oliver implemented this method at scale across all the phenotypes in the portal. And so this method takes into account common variation and rare variation, and comes up with a score and categorizes the evidence, the strength of the evidence that supports this gene being involved in any of these phenotypes. So for example, the evidence is compelling that SLC30A8 is involved in type 2 diabetes. So this is on the top of the gene page currently. And then we have 2 really similar displays, one for common variant gene level associations. These are the MAGMA scores, scores from the MAGMA algorithm across all traits in the portal, gene level scores. And then we have the rare variant gene level association scores that I just talked about a little bit. So it makes the gene page quite long, and it's hard to see what's on it. So now I'm going to show you our development database. And this is what you will be able to see after our release. So we've condensed these in a way that we think makes sense. So at the top now, there's just one section, gene level associations, and there are tabs. So you can either see the huge scores, the common variant associations, or the rare variant associations. And so this makes sense because the huge scores are derived from both of these. And so that we think this makes the page a lot easier to navigate. All right, so next we're going to talk about tissue-specific gene expression, and I am going to turn it over to Annie to do that. Unless there are questions about what I just talked about.
I don't see any questions.
I think we're okay. Okay, good. All right, Annie, take it away.
Thank you, Maria. My name is Annie Moriando. I am a software engineer on Jason and Noel's team. And I was one of the engineers, or I guess the main engineer who developed this new feature that we are talking about today, which I presented at Parliament, which is the gene expression plot. So I will just give you a little sort of overview of what we're talking about when we developed this plot, what our goals were, what we're doing, and then little live demonstration, which is kind of the rerun of what we did at Parliament, but with the benefit of a couple of of months of hindsight. So I think if I just show my web browser, that should work. And what I'm going to do is go into presentation mode, and please let me know if you can actually see, because I don't know if that's going to mess it up. But I like having it in that nice glossy presentation mode. Can you see the slide?
Looks good.
Excellent. Okay, so gene expression. What are our questions that we were trying to answer by doing this, and what are the goals that we had in mind? Now, keep in mind, I'm an engineer. I'm not one of the data scientists or the people curating and processing the data. My role has been to accept the data and turn it into a feature that everyone can use. But based on sort of my and Parul's shared understanding of this project, this is what we talked about at Parliament and what I'm going to revisit for you today. So what we're doing is we're displaying, as the name suggests, gene expression across different tissues and cell types, and hopefully in the future we can add phenotype or disease state. But at the present, we're talking about you search a gene, and that displays graphically for you the tissue and cell type variation in gene expression. And so we've got many different sources: GTEx, Geo, HPAP, Blueprint, AMP Consortium, and also obtained from publications. And at press time, we had 256 data sets, but press time was in March. So I think actually, is that still accurate, Maria? Do you think we've gotten more since then?
I think that is what we have at the moment. We are
Okay.
definitely planning to add more in the future, though.
Okay, we're planning to add more in the future. We're always working on this. And the goal is to aggregate as much data as we can get and sort of harmonize it between all these different places that it comes from. No matter where it comes from, our plot can incorporate that and show you the fullest picture possible and give you the most robust results that you can for the gene that you're trying to look at. So since I'm an engineer, I wanted to speak a little bit about the UX UI concerns. So, okay, what are we trying to do? What is the user of this feature wondering that leads them to this feature? How expressed is this gene in my tissue of interest? That's kind of the basic question, right? What types of samples were examined? And how is the data distributed? And that's really a question that can only kind of be answered graphically, I think. The distribution of something, you want to see a plot. You want to see it before your very eyes. And if you have distribution, results that surprise you, or if everything is clustered together, or you have an outlier, or you have, something that is surprising to you, where's the provenance of that data, right? Chase down whether it's as expected or not as expected, or anything that is of interest to you about the gene expression distribution, that you can chase it down to the source. It's not just a dot on a plot. It's something that you can examine further and say, okay, well, if this gene is highly expressed in this one type of pancreatic tissue, where'd that result come from? And that's where we're trying to answer. You can answer these questions right within the knowledge portal. So what we took to calling the violin multiplot shows you side-by-side results for many different tissues for the same gene. You search up the gene, and then you have parameters that you can set to refine further what you're looking at, but basically all the results for that gene are going to be laid out in front of you. And SOD1, so it's a common one that we like to talk about a lot, You can select a log scale or a linear scale. You can set a floor for if you don't want to look at small data sets, or if you want to really see only data sets that had more than 4 samples, 10 samples, 200 samples. You can filter that so that you don't, these nice large violin plots versus, small ones that you can barely see with the naked eye. And you can filter by collection source. You can sort by the provenance of that data. And this was a walkthrough that I think this is when I was standing on a stage presenting. It's probably easier for me to just click right through to the demo itself. The plot generates automatically. Once you search up your gene, you can set the parameters, view everything that results, and perhaps most importantly, follow all the data sets to their source. And then, okay, so let us go over to, do you see now the portal live on my screen?
Yes, thanks, Annie.
Excellent. Okay. So batch two. I say Bach because I'm a musician, but I might be wrong. We've got Bach 2. And this is the default view. It displays everything in logarithmic view because if you view it in linear, it tends to have sort of bottom heavy looking plots. And we didn't like this. We preferred a logarithmic scale where TPM plus 1, so that, obviously there are plenty of zeros, that would mess up a plain log. But, you're really seeing order of magnitude, right, for how much this particular gene is expressed. And we have all of these different tissue types. You can sort of see a further breakdown within that tissue type by expanding the data set panel in the bottom. But if you set, let's say, a floor of 5, we're only going to see data sets with more than 5 samples in them. Already, you can see that changes it a lot, right? So total samples can be in the double digits or the quadruple digits just by looking at this. Let's say I want to see only data sets with 1,000 samples. Right. Okay. So now this changes the picture pretty dramatically. You're going to see, obviously perhaps a less smooth plot because you're filtering out some of the samples. But, if data set size is something that is important to you, you can view in real time, or you can also say, oh, I only want to see blueprint results. Okay. I guess it's just blood that gave us that. Or I only want to see GTEx. I think it pretty much speaks for itself. I did my very best to make these things intuitive, but I'm curious to hear other people's thoughts. What interests you? What is it that you would be looking for when you come to this project? And also, okay, so here in the data set panel, you can see things broken down. Okay, let's go back to the everything view. You can see things broken down by the provenance of the data right here. And if you decide I want to look at this data set, you can go straight to the source. And yeah, that's sort of what this is what we're hoping to share with the world. And we had in our future directions, we're hoping to obviously add some single cell results. This is a hot topic for us. We're hoping to incorporate things like phenotype or disease state. If you want to, if there's a gene of interest in type 1 diabetes, it might be nice to filter samples from people who have the phenotype of type 1 versus don't. And then to have, more granular bispecific biosample that you can filter in or out of the results that you're seeing, because it's dynamically calculated in real time. That little distribution plot is every time you set your parameters, it recalculates it. So you might want to be able to say, this is my favorite biosample, and only view data that comes from that. Okay.
Just to add to that, Annie, which is great. These are, the reason the future directions are really useful, maybe to hear some feedback from you all about it, is particularly because so much of the data being generated by this community right now is in the single cell space. And so much is also moving towards the disease state in the specific tissue. And that's remarkably useful and powerful, I think, to our user base. So as we pivot in the next 6 months or so, that's sort of the new sort of functionalities coming in from two perspectives. The first being integration of data relevant for the tissues and with single cell based experiments, and then making visualizations for them so that the user can make decisions with that actual data. And that's going to require some different filtrations and visualizations. And the reason we put the gene expression in bulk first is just to give a sense of like where it would be, how it would sort of be part of an overall workflow from the gene page on the portal, as Annie mentioned. And then sort of after that, what's the next step? Because then there becomes more of a visualization of single cell data in the context of that tissue or whatnot. So you're moving away from the genetics. So we want to sort of figure out how to make that workflow. So we're also curious how, as a user, you might like to do that, because that's something we're kind of working through. Specifically, what types of filters and sort of ways to look at the actual results and the U plots and the violin plots that would be valuable to our user base is something we're thinking about. So this is the beginning of that sort of conversation and moving towards workflows in that space. So just to follow up on what Annie said.
There is one question that I think we can talk at this point before we go on. But in the Q&A, there is a question where GTEx has data on more than the tissues that you showed. Do you have a more incorporated IE from the digestive system?
Good question. So we don't have all the GTEx data, you're correct. I think our colleagues at CMDGA preferentially pulled in data for tissues that were relevant to common metabolic disease, because that's what we sort of started with. And so their next activity is to comprehensively integrate GTEx results into the portal, specifically across all tissues that we don't currently have represented. So those are visualized in the portal. So you're right that they're not fully in there, but that's definitely on the docket for the fall.
Another question that we have is, how does this relate to the data in Nomad?
Maria, do you want to take that one?
So Nomad has allele frequency data, as I understand it. I'm not aware that it has expression data, so I'm not sure. I could be ignorant here, but I'm not sure I understand how it would relate. Anyone else want to jump in on that?
So yeah, very different data. So they have sequence data. We do have some known allele frequencies that we represent in the portal. So it's a very different UI, but similar representation of variation, right? I think that's the question. I don't see the question, so I'm trying to figure out the context. If someone wants to unmute themselves and ask it, that would be helpful so I can understand what the question is. Sorry. I'm trying to see the question.
It's in the answered section of the Q&A.
Oh, okay, I'll take a look.
Yeah.
So I'll take a look and I'll answer that offline. So also John Paul, hey John Paul. If there's a filter for the analytic approach, RNA Viz, very sorry for this. There'll be filters for different types. Go ahead, Annie, answer that one, yeah.
No, that's this is exactly why we do these webinars. No, we don't have a filter for analytic approach. I'm not sure if that's on the bioindex. I think it is, but I wouldn't swear to that. But yeah, so if we possess that data, if we possess it all annotated by analytic approach, then yeah, cool. Boom, we can do that if we decide that that's what we want to do. If we don't, if it it is in gene cards. Okay.
Yeah.
Cool. Then we can talk about this as a future direction. I would need to see what we have and see how to incorporate that. But yeah, that's interesting if that's an approach that people want to see. If that's a feature, a parameter that people want to see.
We have two other hands raised. Who should we start with? Eva?
Hi, I hope you can hear me okay. I did send you quite a bit of notes. The world of intestinal reactions to food via the hormones GIP, GLP, you see a lot online. There's a whole lot of research that still needs to happen. And so in terms of type 2 diabetes, I believe we're going to have to wait maybe 6 months before people should expect to know more about that particular area. I see the research going that way via Twitter. I see it via my own, focused attempts to try to figure out all those hormones. And so, but yeah, once we get there, then we might be able to add more. Someone asked you about that particular part. The portal will be very, very helpful once the data comes through, but I can see the biochemistry and research is just now starting to happen. So thank you.
It's a great point, Ava. I think for us, we're thinking about how to incorporate relevant data like that going forward. So I'd be curious to talk to you when that sort of comes about, and we can figure out how to integrate things like that into the portal. I see another hand raised.
Thank you. I would like to, I'm Inji from Yanxi and Genji. I'm so excited that you are keeping making steady progress in such efficient way in this portal. I would like to take the chance to ask Maria a relevant question for the gene-based web page. So I'm just curious. When we browse the gene page, we notice, wow, there are some circumstances that the gene-based aggregate test based on whole exome sequencing results are significant, while sometimes the SNP-based result from G was not significant, or the other way around. And in rare cases, in rare instances, that we got contradictory results from gene-based aggregated result from whole exome sequencing versus SNP-based GWAS results. I would like to just wondering how to take a step forward to understand the biological mechanism. How can we reconcile the result we get from available out there from gene-based test versus SNP-based results? Yeah, I appreciate your comments. Thank you.
So Yingji, your sound is a little bit garbled, so I had a lot of trouble understanding the questions. I understand you're looking at some results that may be conflicting in the gene-based tests, but I'm afraid I really didn't get the whole gist of it. If you could email me, I would be happy to look into that and answer you. Okay, thank you.
Thank you so much. Thank you.
Okay, and I know before we leave the gene expression topic, DK wants to say something. DK, do you want to just say it yourself about the violin plots?
It's a pretty simple thing. I mean, so Annie has developed the feature, the plot and the table, not only for the portals, but also the BEOR service, BEOR platform we offer for free. So it's not officially released, but when it gets released, you'll be able to actually use the plot and the table on BEOR. Against your own data.
That's awesome, DK. So what DK is indicating is, as you know, we have, as a result of building portals, we've been very lucky to build a software surface in addition to that, so that users who contribute data, who want to make like a single page for their publication, or want to have something private as they do some work and analysis with us, and then we make it accessible in the knowledge portal, we built this framework called Bring Your Own Results, or BEOR. And what DK is mentioning is that software package is available as part of our resource, but also keeps evolving as we add more features to the portal. We make those visualizations available in BEOR for you to make your own. But then vice versa, as we work with you all in this community to represent different types of omics data, perturbational data, screen data, other complex GWAS association results, we've been making new visualizations and tools there, and then those go back into the portal. So it's a nice complementary activity. So if you actually go to our homepage, it's on there. You go under see more portals, you can get there, and also it's available on right at the bottom of the homepage on any portal, so you can get to the page to make all your BEOR portals, and also reach out to us if you want to make them.
Okay, and I see we have a couple open questions about new data types, and I would just say, yes, we're going to include them. So Christina asked, are we going to include DNA methylation data? John Paul asked, are we going to include EQTLs, PQTLs? Yeah, those are all data types that we can take in through our sister resource, CMDGA, and then display in the portals. Those are all, I think, in the future plans. We don't have them quite at the moment. John Paul also asked about miRNA. And we are also in the process of adding annotations for non-protein coding genes, so non-coding RNAs of all types. So that will be available on the portal fairly soon too.
Yep. And just to add to that, Maria, I think CMDG already ingests DNA methylation sites and represents those, both the raw repository data and sort of visualizations of those. So those are available already on CMDGA. We just haven't incorporated them in terms of our visualizations incorporated with the genetics. So that's nice. In terms of PQTLs, MQTLs, and things like that, those are on the docket for the fall as well, because we finally have like a very clear data set from a collaborator where we're gonna work together to figure out how do you actually visualize that in the context, again, of human genetics and the sort of the flow and the workflows that we have on the portal. That's our sort of next move. So that's a great question to raise, Jean-Paul. Thank you.
Okay, great. I'm going to go on to do a demonstration of the new gene finder, the updated gene finder. So let me go. So to find the gene finder, you go to tools. Let's see, we're in the development. Okay, gene finder. And so this diagram up here shows what the current gene finder in the portal does right now. And it's fairly simple. It lets you choose traits, as many as you want, actually. And then it looks at the common variant gene level association scores that are generated by MAGMA. And it will filter the genes such that you come out with a list of genes that are associated with all of the phenotypes you selected. And that's really all it does. And so the new updated gene finder will have this still, of course, but a lot more. So let me demonstrate. So again, you will start by putting in phenotypes that you're interested in. So we'll do, let's see, I have to move my window up a little bit. There we go. We'll do T2D and BMI. So we've just selected two phenotypes, and it is loading a table and a graphic of the results. So the current gene finder has a sort of a Manhattan plot of all the gene level scores, but we decided that it wasn't really all that relevant to know where in the genome a gene is located. That's easy to find otherwise. And so DK's come up with a more easily intuitive, sort of more intuitive and quickly interpretable graphic that shows a lot more information. So once you select your phenotypes, you get this table, and you get some extra information along with. So here's a common variant gene level score, say for this gene for these two phenotypes. But in addition, you'll see the huge score, the Human Genetic Evidence Calculator, for each of these phenotypes. You will see a p-value for the rare variant association. But the caveat here is that, first of all, it's only shown if it is significant. Well, if it's less than 0.05, which is, yeah, so if it's not shown, if it's larger than 0.05, it's not shown here. And also, we don't have the rare variant gene level scores for all phenotypes. So that may be why it's not here. But where it's available, it's shown. And then we have things like the number of samples and the combined p-value across all of the phenotypes that you've selected, and a link to view this region in the variant sifter if you want to go on and explore that way. But you can add a lot more information to this table now. So the tissue-specific gene expression that Annie just showed us, you can add those values to this table. So you can choose a tissue category, and then the values for each gene in that tissue will be added. This is transcripts per million, and then the total sample size. So you can see that for each gene, and you can choose as many tissue categories as you want and see the values for all of those tissues for each gene. Then you can also add more information. You can add whether these genes are present on lists of predicted effector genes. So I've talked about these lists before. These are just lists generated by researchers who want to predict the most likely causal genes for a disease or a phenotype. And we're collecting and curating these lists and representing them. And so here you can add, let's see, I know we have a list for obesity, so let's find that. We'll add that. And then I'll add our original effector list for type 2 diabetes. And I know in this case that the first author is Mahajan, so I will start typing that and add that list. And now that we've added that, we can see in the table that which genes are on which lists. And if you mouse over a list name, you can link to the original publication if there is one, and also just go straight to see that list. So already this table has a lot more information than it used to. And the graphic represents all this in a nice way. So here, the gene level scores are shown in the shapes at the top. The green represents the huge score in different shades. The gray circles represent the common variant gene level scores in different sizes for different significance. And then red circles represent the rare variant scores. And again, different sizes for different significance. Then the expression level in the tissues you selected is shown here and also on mouse over. And the presence of the genes on of each gene on these effector lists is also shown graphically. So, and once we've generated this table, we can filter the results by a lot of criteria. So if you want to restrict the table to genes that have a certain huge score above a certain thing, you just type in a threshold here, and then the table will update to show only genes where at least one of their huge scores for one of the phenotypes is that value or higher. And you can do the same kind of threshold filtering for the common variant p-value, the rare variant p-value, and the transcripts per million of gene expression. You can also filter the table so that it shows only the genes that have rare variant results, for example, and only the genes that are present on the effector lists that you've chosen. So you can really narrow down the table in a lot of ways. Finally, this little link over here lets you sort of customize the way the table looks. You can set a threshold for common or rare p-value, and it will change the highlighting on these blue boxes to highlight everything that is more significant than your threshold in blue. And you can also hide and restore columns of the table if you just don't want to see these p-values. So we think this is going to be really useful, and we think it's just the start of adding even more versatility to this tool. We think it's going to be a great way to just explore many of the different data types we have and find genes that answer many custom criteria. So let's see if we have questions on this interface in particular.
One thing I would like to add.
Yes, please.
Maria's. So if you scroll down just above the table, you have download button. When you click it, it's going to download the data. I mean, the data you see on the table in CSV format. So you can load the data and then filter the data by filters, and then just click the button and you get the filtered data on your local machine.
Thanks, DK. I meant to mention that and forgot. And let's see if we have more. I don't think we have any other comments, questions just about this. So great. Okay. Now let's see. I wanted to go back to the slides. Okay, so I wanted to talk just a little bit about some future plans. So as I think you know, we have this tool called the Signal Sifter, which lets you do a sort of a similar thing to the gene finder. You can choose a trait. So you start with one trait. You get variants now, not genes, variants that are associated with it in clumps by linkage disequilibrium. Then you can choose more traits. And what you end up with is a list of genomic regions where there are associations for multiple traits. And so working on the gene finder and then thinking about this interface and also talking to colleagues got us thinking that we could really augment this interface. And so this is just a screenshot of the type of results you get. You see the clump is the genomic region, and these are the variants and their p-values that are associated with the different phenotypes that you select. But we realized we could really broaden this tool into a signal finder that we think would be quite useful. So say the user wants to just look across the genome for all regions that are significant for a given trait. We could then let them filter this list by many other criteria. For example, the posterior probability of association for variants that are included in credible sets. Of course, we could add more traits and do that across those traits. We could also look at overlap of the variant with an epigenomic annotation, promoter or enhancer, something like that. And we could even add more filters like links that are generated by 3D chromatin conformation experiments that say that a variant is in contact, for example, with the promoter of a particular gene. And other criteria, like if a gene's on an effector gene list, and et cetera, et cetera. So yes, the end goal of this would be a list of regions that the user might be most interested in. And then of course, links to the variant sifter tool for each of those regions, which would allow them to explore in great detail all of these data types. So we're just really starting to think about this, and we would love to have your feedback on, whether something like this sounds useful and what you would like to see it be able to do. Okay, and I'm going to just briefly talk about another future-looking thing that we're working on. So I've mentioned it today and previously a lot about these effector prediction lists. So this is just an example of one of them, but the bottom line is that people take a GWAS, look at the significant loci, integrate a whole lot of different methods, and then predict which is the causal gene at this locus. And so we've been looking at a lot of these lists, trying to identify them in the literature and curating them. Oops, sorry, I didn't mean to advance there. So there are more and more of these in the literature, and they can be hard to find. UA is helping us to find these papers with her automated literature searching skills, but it's still pretty difficult to identify these papers. And there's just no standardization. There's no standard way to share these or report them. There are no standards for data type or evidence. And they're very different from each other. And many of them are just kind of hidden in supplementary data. So we think it's really time that the field talks about this and comes up with some standards and conventions. And so what we're hoping to do is to actually organize a workshop to bring people together to discuss this. We're talking about doing this sometime in the fall, and we're just working on plans. But we would like to have just a one-day workshop where we talk about all these issues and come up with some conclusions and write up, a white paper or a publication that outlines these conclusions. And we think this would really be valuable for the whole community.
In particular, if anyone's in this call is interested in participating, please send an email to me or Maria or to our help email, because we are actively looking for session leaders and folks who are really passionate about this to participate. The GWAS catalog was really successful when they did early in the days of GWAS standardization. And so we decided that, in concert with them, that we're going to take this on as far as a sort of a community effort where we actually might provide standards for others who are going to build these lists in the future to make it super easy for them to represent them, catalog them, track the provenance, but also, how to share them with each other. So, that's what our objective with this is, because I feel like this is an incredibly valuable tool as we as human geneticists can give to folks who are not human geneticists. The reason I say that is because so many human geneticists think about things in Manhattan plots and, p-values and direction and effect size, right? And that's great. And that teaches us a lot. And our resource is very good at sharing that. But the community has moved, right, to layer other evidence sources in terms of taking these GWAS loci and prioritizing a specific gene within those regions, presenting that knowledge back to the community from publications and from curated knowledge and from work that we're doing ourselves, I think is really informative for our human genetics community, but also for a basic scientist who's like, I just want to know what are the genes from these, 500 signals that they have for cardiovascular disease. What are the ones I should be focusing on? What are the genes and why? So that's really why this is, I think, a very valuable tool and really pivotal to the work that we want to do as part of this resource.
Great. Okay, so I think that's it for me. I just have a brief slide on upcoming things. If anyone's going to the American Diabetes Association, Mackenzie and I will be there. We're running booth number 841, and we would love to have you stop by and talk to us. That would be great. Other than that, our next focus group is going to be run by our colleagues at the University of Michigan, Laura and Ryan, who are working on a nice visualization for co-localization of EQTLs and GWAS loci. So that should be really interesting. And then our webinar in August will be Patrick Seal from the University of Pennsylvania giving a scientific presentation.
Great. I have a couple of comments before we wrap up, but any questions folks have? I don't see hands raised.
Yeah, and there are some in the chat. Some of them I can
answer offline.
So I'll stop sharing and you can talk.
Sure. So so that was great. I hope folks found that useful. I think we haven't done this in a while. I think in terms of the co-locus, please tune in for that. That's going to be really great. We have so much data in that space. So many users want that visualization presented alongside of what we currently do. So please tune in for that to give us feedback. And Laura and Ryan have been doing an incredible job building that UI and adding the relevant data to it. So that'll be really exciting. A couple things actually useful as you think about the broader resource. Just I want to add in the last 2 minutes. I'll just quickly share my screen if I can. I think I can, Mackenzie.
You should be able to.
Yeah, look at that, just like that.
Yeah, there we go.
Nice and easy. All right, super. Maria covered some of the things I wanted to cover too, which is great because great minds think alike. But you know, one thing as you're using the resource, that if you're not interested in the UI, which many people are, but if you are a computational a specialist and want to download all of the matrix of the index results, just so you know, that's all available to you as well. It's called our bioindex. It stores all the results and representations of the relationships that we calculate across all the bottom line results, the VEP annotations, the LD score regression results, anything you want essentially in terms of the different types of results that are represented in the portal in visualizations and tables. You can also get programmatically, and it's through this bioindex. It's not fully exhaustively documented yet because there's lots to document, and so we're working on it, but it's there. It's pretty well designed by Patrick and others on our team, and pretty clean and clear. So I think you can navigate it if you've used any APIs. But what you can see right here is you can search the different types of indices we have for you, and they're pretty logical. They actually mirror very well the different results in the portal, but there'll be some documentation. But if you're so inclined and intrepid and want to access all of the matrix of our index results, you can get them here or contact us if you want to help to get specific queries done for you. So that's just, I thought, really important we've had many people in the user base who want to look at that specifically. So Maria mentioned our future events. Particularly ASHG is a really important goal for us because that's another time when we will be releasing a new version of the Association to Function Knowledge Portal with a lot of the upcoming data sets, particularly focusing on credible sets under annotations and data types in the single cell arena. So look for that. Also look for improved sort of representation across all the common disease areas for our ASHG release. So that'll be in early November this year. So hopefully we'll hear from you then. Again, please reach out to us. I also popped into the chat the BIOR link, which I think is really quite relevant to the conversation we have today for folks who have other data types they'd like to represent and share on some visualizations that maybe we would contribute to the broader portal, but particularly just for you to have and for you to use in your manuscripts and whatnot. So if there's no more questions, I think we're going to end right on time. Did I get that right? And here's, again, the team that makes this all possible. Thank you, Andy, for presenting as well, and DK. It's always great to have our engineers here because they think about the resource in a better and great perspective. That's really nice to have. And contributors to our disease communities, and these are experts who give us results, data, and more importantly, community feedback of what's really important there for their specific disease communities. So with that, I think we'll end. Yes, Maria, any wrapping comments you have?
I think that was it.
All right.
But
Thank you, everyone.
Get in touch with us if you have questions, or more things to talk about. We're always happy to do that.
Thank you.
Bye-bye.
Bye-bye.